I am Dr. Khokon Kumar Dotto. My Christian name is Dr. David Boone. I'm currently working as assistant professor in the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University in Gopalganj, Bangladesh. The title of my discussion is T-Xnip gene therapy shows promise in preserving vision in retinitis pigmentosa patients. The retina is located at the back of the eye. The thin layer of tissues of the retina consists of rod and cone photoreceptors that convert light into electrical signals that the brain interprets as vision. Retinitis pigmentosa is an inherited disease of the retina. It is a progressive degenerative condition that can lead to low vision and even complete blindness. More than 20 million people are currently suffering from retinitis pigmentosa worldwide. In Bangladesh, retinitis pigmentosa contributes largely for a huge number of blinding cases. In the USA, more than 100,000 people suffer from this rare disease. In Denmark, the frequency of this inherited ocular disease is relatively high than other countries. To note here that retinitis pigmentosa can develop in other eye disease also. At present, there is no cure for retinitis pigmentosa, but the doctors usually try to use some options to slow the vision loss and in some cases even to restore the sight to a significant extent. Rods are in the outer side of the retina and help us to see in dim and dark light. Cone photoreceptors reside in the central locations of the retina and help us to perceive fine visual details, color, and high acuity vision. In the early stage of retinitis pigmentosa, rod photoreceptors are more severely affected than the cones. When people experience night blindness and the gradual decrease in the visual field. The visual field is the encountered visual area without moving the eyes. In the late stages of the retinitis pigmentosa, the cone photoreceptors die and the patients lose the daylight vision. In the late stages of the retinitis pigmentosa, the patients experience difficulty in performing essential tasks of daily life like reading, driving, walking without assistance, or recognizing faces and objects. Retinitis pigmentosa is a rare inherited disorder and a harmful mutation in any one of at least 50 genes seem to act as the causative. The harmful mutations related to retinitis pigmentosa are transmitted in the offspring in different ways like autosomal recessive inheritance, autosomal dominant inheritance, and X-linked inheritance. Very recently, a landmark scientific advancement towards the successful treatment of retinitis pigmentosa has been reported in the scientific journal. Yanlu Zhu et al. Published an article in Elife on April 13, 2021 entitled AAV-Txnip prolongs cone survival and vision in mouse models of retinitis pigmentosa. The group screened about 20 retinitis pigmentosa related genes for the rescue capabilities of cone photoreceptors. The rescue effects was the best for thioredoxin interacting protein TXNP. The group used a harmless adeno-associated viral vector for in vivo delivery of the gene. The rescue effects were greater for a mutant type TXNP C247S in compared to the wild type TXNP. When amino acid cysteine is replaced with serine at the position 247, TXNP cannot stop the antioxidant activity of thioredoxin. But TXNP C247S preserve the functions of wild type TXNP in glucose transport, glucose metabolism, lactate catabolism, regulation of genes transcriptions inside the nucleus that are involved in the mitochondrial structure and mitochondrial functions, at least in energy metabolism. TXNP C247S gene therapy helped the cone photoreceptors with the ability to select alternative fuel resources, and at the same time, the mitochondrial health was greatly improved. The rescue effects of TXNP C247S was dependent on lactate dehydrogenase B and lactate catabolism. Three things are widely accepted by the researchers as the causatives of cone death in retinitis pigmentosa: oxidative damage, inflammation, and a shortage of nutrients. The group found a combinatory approach to work much better when an additional gene therapy that reduces oxidative stress and inflammation was administered along with TXNP C247S gene therapy. High level of protection was found in the cells. Major part of the intracellular ROS are generated inside the mitochondria. High level of ROS generates oxidative stress. When wild type TXNP translocate inside the mitochondria, it limits the ROS neutralizing activity of thioredoxin 2, which is only found inside the mitochondria. ROS is also essential element in mitophagy, inflammasome activation, and subsequent inflammation. It is also known today that TXNP directly interact with NLRP3, which is a vital component of inflammasome complex. To note here that Khokon Kumar Dutta et al. For the first time reported the presence of TXNP inside the mitochondria of rat renal tubular cells in 2005. We also reported for the first time internationally that TXNP is a binding partner of mitochondrial TRX2. We also suggested the possible involvement of TXNP in the regulation of Warburg effect or lactate metabolism. Our research works on TXNP was published in 2005 in Nature Laboratory Investigation with the title Two Distinct Mechanisms for the Loss of Thioredoxin-Binding Protein 2 in Oxidative Stress-Induced Renal Carcinogenesis. Anyway, now we need to wait to see the advancements to ensure the safety and efficacy of the gene therapy using TXNP in different other animal models, which will give voice for its successful translation in human benefits. Finally, I would like to wish good health and better days for you everybody. Thank you.